Lipids are compounds that are soluble in organic solvents such as acetone, ether, and chloroform. The term lipid, derived from the Greek word lipos, meaning fat, is a general name for oils, fats, waxes, and related compounds. Oils are liquid at room temperature, whereas fats are solid. Lipid molecules contain the same structural elements as carbohydrates: carbon, hydrogen, and oxygen. Lipids, however, have little oxygen relative to carbon and hydrogen. Fatty acids have a carboxylic acid group at one end of the molecule and a methyl group at the other end, separated by a hydrocarbon chain that can vary in length. The carboxylic acid group can bind to glycerol to form a mono, di, or triacylglycerol. Although the solubility of the different lipids varies considerably, they generally are seen to dissolve poorly in water. The three classes of lipids most commonly recognized are simple lipids, compound lipids, and derived lipids. Triacylglycerols, or triglycerides, are the most abundant dietary lipids that are consumed by humans. They are composed of a 3-carbon glycerol backbone esterified with 3 fatty acids. Triacylglycerols differ in their fatty acid composition, and this is covered in more detail within your written lesson titled Lipids. When we are reading or learning the science of nutrition, we are likely to see a lot of different abbreviations, and this is important to keep in mind as we go through this next bit of information, because we're going to talk about fatty acid chains, and these are broken down into several different types. So in humans, the chain length of fatty acids typically varies from C14 to C24, with C representing that carbon backbone that determines the different length of these fatty acid chains, although there are shorter or longer chains present. Fatty acids with the chain length of C8 to C10 are considered medium chain fatty acids, or MCFAs, and those with the chain length of C6 or less are considered short chain fatty acids, or SCFAs. The most abundant fatty acids are the long chain fatty acids, or LCFAs, which have a chain length of C12 or more. Fatty acids with no double bonds in their hydrocarbon chains are called saturated fatty acids, or SFAs, and those with one or more double bonds are unsaturated fatty acids, or UFAs. With those abbreviations out of the way, let's go ahead and move on into something you're probably more familiar with, which is cholesterol. Cholesterol is a lipid found in the cell membranes of all animal tissues, and it's transported in the blood plasma. Cholesterol is considered a sterol and is really important in the function of the cell membrane, where it regulates membrane fluidity over a wide range of temperatures. Cholesterol aids in the manufacture of bile, and it's important for the metabolism of fat-soluble vitamins A, D, E, and K. Cholesterol is also the major precursor for the synthesis of vitamin D and various steroid hormones. In terms of function, lipids are a really important energy source, especially during prolonged exercise. Large amounts of fat can be stored in the body as well, mainly in the subcutaneous adipose tissue, from which it is mobilized and then transported to the organ that uses it. Skeletal muscle contains a directly accessible storage of fat as intramuscular triacylglycerol. Lipids have many other important functions, including it being a fuel usable for most cells in the body and an important fuel for contracting muscle. Fat protects vital organs such as the heart, liver, spleen, kidneys, brain, and of course the spinal cord. A layer of adipose tissue covers these organs to protect them against trauma. Now, about 2 to 4% of total body fat is stored around these vital organs, and the intake of fat-soluble vitamins A, D, E, and K, as well as the carotenoids, depends on daily fat intake, with fat being the transport medium within the body. Phospholipids and cholesterol, which we've already mentioned, of course are important constituents of cell membranes, and we also remember that cholesterol is an important precursor in the formation of bile and is itself an important component of bile, but we'll also now see that it carries on a really important function, especially for men. We have stated that cholesterol is a precursor for hormones, but now we're going to be more specific and say that it's a precursor in the synthesis of steroid hormones or sex hormones. So the synthesis of testosterone or progesterone, for example, and among others, relies on cholesterol. Linoleic acid also plays an important role in the formation of eicosanoids, which are hormone-like substances formed within cells serving a regulatory function. And related strictly to intake, we know that fat will usually make food taste better or seem more appetizing, but it also has many aromatic properties and substances that can stimulate appetite as well. Only some of the lipid forms that we consume can be used as a fuel, and these oxidizable lipid fuels include fatty acids, intramuscular triacylglycerols, or IMTGs, and circulating plasma triacylglycerols, which would be chylomicrons and VLDLs. And VLDL is the main transport of triacylglycerols from the liver to adipose tissues and muscle, while HDL, or high-density lipoprotein, is transporting cholesterol from the peripheral tissues to the liver. Therefore, chylomicrons and VLDL may play a role in energy metabolism during exercise, and HDL is not believed to play a significant role in energy provisions for muscle. In addition, fat-derived compounds such as ketone bodies can be made and then serve as a fuel whereby glycerol can be converted into glucose using the process of gluconeogenesis within the liver. High fat intake along with low fiber intake has been linked with increased incidences of colon and prostate cancer as well as increased body weight. Studies have suggested a correlation between high dietary fat intake and obesity, and while that seems to make sense, there are plenty of other studies that seem to raise questions about the effect of the percent of energy from dietary fat on our body weight. Therefore, the evidence that high-fat intake contributes to obesity is actually insufficient to be able to make a solid or definitive recommendation for a very low-fat diet. And as a result, most guidelines for Americans state that fat intake should be moderate instead of being defined as low. Now, a very low-fat intake paired with high carbohydrate intake could actually have adverse health effects on a client. Reducing dietary fat to below 20% of total intake has been seen to elevate plasma triacylglycerols and also is seen to increase LDL cholesterol while decreasing HDL cholesterol. And this lipoprotein pattern or cholesterol pattern or profile has been shown to predispose one to coronary artery disease. The associations between high fat intake and cancer may be due to the intake of saturated animal fats and not from vegetable fats, raising the possibility that fat totals per se are not the only important factor for us to evaluate. Fatty acids from fish, on the other hand, seem to protect against cancer, so as you can see, research and science continue to redefine our perspectives on fat intake. In summary, our body burns what is available. It's like the fuel in your car. It will burn whatever gas you put into it. If a high-fat meal is consumed, for example, there is more fat in the system and more fat is then burned. However, this does not imply that we burn more body fat. Available fuel from fat and body fat are different. Likewise, if a high carbohydrate diet is consumed, carbohydrate provides more fuel during exercise. Since our body stores a limited amount of carbohydrate as glycogen, it is fairly quickly used up since the body continues to work, even when at rest. So it becomes important to remember that fat is a secondary source of fuel, but there is a lot of misinformation out there for our clients to read and sort through. Regardless, the bottom line is that if glycogen stores are low for our client during an exercise session, there is usually more fat available for fuel during exercise.